Thanks a million, Peter. Mitch, really appreciate the invitation here. I'm standing here in part because of the influence of Bob Comis. I was one of the people that benefited from his mentorship over the years and the opportunities he created for me to further my career. So thank you, Bob. I'll make a little confession here. I'll always view myself as a clinical investigator, clinical trialist, even though now I live in the world of big data and observational data. And I'm going to try and sort of weave a theme through in 14 and a half minutes that will connect these two worlds in a way that hopefully we can leverage the opportunities in both together to advance drug development and ultimately cancer care. So without further ado, to level set in terms of what we're talking about with real-world data, there are many sources of real-world data, and I'm going to focus on data from electronic health records, but this slide is just to highlight the fact, as Larry mentioned earlier, there are other sources like claims data, registry data, and data from devices that patients may wear or enter information from their homes that make up the whole universe of real-world data. And then simply put, real-world evidence is the evidence derived from the analysis of real-world data. So you got to have like high-quality data upon which to generate evidence. Many of us grew up in worlds where the notion of observational data actually being useful to make clinical decisions was looked down upon. The reality is that real-world evidence isn't new. This is just to highlight that the treatment of scurvy with vitamin C was not based on a prospective randomized clinical trial. That's the chemical structure of insulin. Likewise, huge benefit, well-known problem. You didn't need a randomized prospective clinical trial to prove these things. And so there's always been this recognition that real-world evidence is fit for some purposes, but it was really very limited, in part because of the quality of the data that existed historically. But a few things have changed, and so this is the slide that explains why Flatiron exists, basically, and other entities like Flatiron. So back in 2009, the Affordable Care Act, American Recovery and Reinvestment Act, had part of it was high-tech, which incentivized the adoption of basically paid people to adopt electronic health records. As a result of that, every cancer patient in the United States essentially has their clinical course chronicled in an electronic health record. There's now this huge demand based on all the science you've heard today and the increasing frequency of certain types of cancer associated to the aging of the population, obesity and so forth, that raises so many questions about new drugs either developed by themselves or in combination that simply outstrip the ability of the clinical trial system to answer all of those questions. And in many cases, the feasibility of doing prospective clinical trials is not there, either because of the rarity of populations or ethical considerations related to equipoise. So there's this huge demand. And then we have on top of that policy in the 21st Century Cures Act that has told the FDA, you guys got to figure out how to use real-world data in your decision-making regarding regulatory decisions. So supply, demand, and policy have all conspired to create an environment where there's a lot of really deep thinking about how we can get real-world data to be of high enough quality upon which to generate evidence and to accelerate cancer care. So To that end, I'm going to take us now back to the electronic health record. It's really messy stuff, as you've heard earlier. The electronic health record has structured data and unstructured data. The structured data are things like laboratory tests, chemotherapy administrations, stuff entered in drop-down fields. But in cancer care, so much of the patient's trajectory is captured in unstructured notes, physician notes, pathology reports, documents that are scanned in from outside laboratories, discharge summaries from outside hospitals, and all this stuff goes into the totality of the electronic health record. And in order to get that information out, it's not trivial. And you heard one of the solutions that Larry was advocating for, which is this notion that we should do a better job of entering key information as structured data. That would certainly simplify things a lot. But now we have a gamish of structured data, which needs to be harmonized and normalized into common data models, and then unstructured data, which still requires, even with the assistance of machines, to search terms, to apply machine learning, to identify cohorts, still requires people in part to read documents and interpret them in collaboration with the assistance of machines. This can allow you to create common data models, a real-world evidence database, and then that can then be analyzed by itself, but also linked with outside sources of data, such as those in genomic databases that aren't necessarily part of the clinical record of patients. They may be part of research databases. There are other things like mortality databases, because the information in the electronic health record only incompletely captures mortality. And then after this linkage takes place, you have to be very careful and intentional about the types of analytic approaches that are applied to these data to account for some of the hazards associated with retrospective observational data. So once you create one of these databases, you can interrogate it in a variety of ways to generate evidence. This is like a snapshot of our non-small cell lung cancer advanced disease database, 57,000 patients. You can pull out demographics on a regular basis. These are updated on a monthly basis. You can look at, for example, PD-1 biomarker, PD-L1 biomarker testing over time since the introduction of checkpoint inhibitors, and look at the impact of drug approvals and new evidence as it's come along in terms of practice patterns. And then you can look at the actual administration of associated treatments, and in this slide, simply showing that over the course of time, the use of checkpoint inhibitors in non-small cell lung cancer has moved to earlier and earlier lines of therapy. But of course, this is only half the story. Maybe it's only a quarter of the story, because what really matters are the linkages of genomics to treatment to outcomes. And outcomes are not typically captured in a very standardized fashion in electronic health records. The sort of things you take for granted as a clinical investigator in terms of a tumor response or evidence of progressive disease, because you have CAT scans and you put calipers on them and you apply RECIST criteria. Those sorts of things don't exist in electronic health records, and new types of variables that can be applied in the real world to electronic health record data need to be developed and validated against gold standards. So as an example, I mentioned earlier that mortality is incompletely captured in electronic health records. And as you can see here, when you compare to a gold standard of the National Death Index, for mortality, if you just look at structured EHR data, you only get a sensitivity of about 2/3 against actual mortality. So if you conduct an analysis based on generated Kaplan-Meier curve, based on structured data from electronic health records, it's going to be wildly inaccurate. So what it requires is, as an example, we link structured data from electronic health records with an obituary, a commercial obituary database, along with Social Security Death Index data, and then further have human abstraction of the chart to see if there were any phone calls or nurses' notes documenting a death event. And ultimately, you can get to a level where the sensitivity and specificity for a death date are at least 90%, so something you can sort of hang your hat on in terms of doing analyses. So what does that enable you to do? Here's an example from a recent publication. This was a collaboration with the FDA, where we looked at in non-small cell lung cancer the relationship between percentage of staining for PD-L1 with both survival and what we call real-world progression. So progression as denoted by a physician saying the cancer is worse. And showing a relationship between the level of staining and the patient outcomes in this data set. As a cautionary note or things to be aware of, data quality is always an issue with electronic health record data, as you might imagine. And so in considering whether the data are fit for a particular use case, fit for purpose, these are the types of things that have to be considered. And I'll highlight a few of them. The idea of provenance, in a clinical trial, when you have a monitor come in and look at every data element that you have collected for a clinical trial, there's complete provenance back to the source document. And so we've set up a system where any data element that appears in one of our data sets can be tracked down to the source, so that the FDA, for example, could come in and ask questions about how did where did this particular data element come from? The idea of measurement reliability and validity is particularly important if you have abstractors who are looking at charts and trying to interpret medical records. And then the validity is particularly important as applied to endpoints, clinical endpoints, if you're going to try and hang your hat on them in doing either comparative effectiveness analyses or, as I mentioned before, the sort of biomarker outcome analysis. Really important from an analytic rigor standpoint to do things that avoid the hazard of cherry picking, for example, in terms of pre-specifying cohorts, pre-specifying the analytic plan, using advanced statistical techniques to address potential confounders and biases in these analyses. So with that sort of background in mind, I just want to talk about a few potential use cases for data from electronic health records. The first one being a precision medicine use case. This is from a database that is a linked data set of clinical data from across the nationwide Flatiron network, linked with Foundation Medicine genomic data on the patients who had a Foundation Medicine test as part of their routine clinical care. And in this case, showing that the PD-L1 status was not related to tumor mutational burden. However, tumor mutational burden was associated with the probability of survival in this non-small cell lung cancer data set. In terms of other potential applications of data like this, one can assess the impact of policies, national policies, local policies on patient care, patient outcomes. This is a collaboration with investigators at Yale University, looking at the question of timeliness of cancer therapy in the face of Medicaid expansion related to the Affordable Care Act. And so we undertook a comparison of patients who were living in states that had Medicaid expansion and those that did not, and asked the question about how quickly after diagnosis patient received their first systemic therapy. And as shown here, and the full presentation can be pulled off the ASCO website. This was a plenary presentation last year, showed that there was a racial disparity in place before Medicaid expansion and in states that did not have Medicaid expansion, which went away completely after Medicaid expansion was implemented. As another example of policy implications, in the middle of 2018, the FDA sent out an alert based on emerging data that the utility, that patients with advanced bladder cancer should not be receiving checkpoint inhibitors if they had low PD-L1 expression. And what we were able to do, and this is a collaboration with Ravi Parikh at Penn as well as Sean Cozen at the FDA, looked at whether there was actually a change in practice that followed this FDA warning and change in the FDA label. Turns out that the FDA doesn't monitor the effect of their proclamations of their approvals or withdrawal of approval. And so this was the first time in oncology where the impact of an FDA narrowing of label was looked at. And as you can see here, in the period immediately following the announcement, the use of immunotherapy and first-line therapy went down. Use of chemotherapy, platinum-based chemotherapy, went up, and the proportion of patients who were having PD-L1 expression testing went up. And so the good news is that oncologists pay attention and tend to do the right thing rapidly after evidence comes out. Now, in terms of clinical trials, lots of opportunities for real-world evidence, real-world data to support the design and conduct of clinical trials. And so this is sort of like a teaser and food for thought for this group. In terms of design, real-world data can be used to help select inclusion-exclusion criteria, to assess the feasibility of conducting a study, of generating a population for study. Can be used to determine the generalizability of clinical trial results that have been reported. They can be used to select sites based on the population at the site. Can help with trial matching to assist with enrollment. You can use the infrastructure that's set up for passive data capture, if you will, to capture data elements on a clinical trial, to not require CRAs to manually collect data from the electronic health record. And in addition to that, you can do centralized monitoring rather than having monitors have to go out to the sites at a regular cadence. That could be much less common. And then, of course, follow-up after the discontinuation of therapy when the patient is maybe not coming to the office as frequently could certainly be done with a data collection infrastructure that is linked to electronic health records. There's a lot of interest now about control arms, and are there settings where a synthetic or external control arm using real-world data could be of use to accelerating drug development. Real-world external controls could potentially be used to assess the treatment effectiveness where randomization just simply is not feasible or unethical. A contemporaneous and relevant comparator arm could help contextualize the result of a clinical trial. Many clinical trials take years to complete, and the standards of care change over time, and real-world data can help understand the relevance of the findings. There's a potential to do sort of a hybrid type study design where you can minimize the number of randomized patients to a control arm and supplement them with real-world data. And all of these things could potentially lead to faster access of new treatments for patients. I'm going to just quickly run through what a hybrid control design might look like. You could start out, and this is just a hypothetical scenario, where you do a one-to-one randomization as you would otherwise do, but enroll, if you will, or identify a real-world cohort at the same time. Assess at a predefined time point how closely that real-world control arm mimics the actual randomized control arm, and if you've done your work well and it looks pretty close to the randomized control arm, then perhaps you can, in an adaptive fashion, alter the randomization ratio of experimental to control patients, supplemented by those real-world data. So I put that out there as a teaser. And then finally, just as sort of like a potential future vision, you can imagine a scenario where a real-world data collection infrastructure is employed both retrospectively and prospectively to allow the select collection of extra data elements, say circulating tumor DNA, extra scans at regular time intervals that would require consent, but layer that on top of the routine collection of real-world data such that the burden on patients, on consenting physicians, and on the data collection infrastructure is minimized in that way, again, making possible sort of faster, cheaper clinical trials. So I'll stop there. Thank you.